So another compare and contrast slide. We're going to look at comparing electron transport and the complexes that we have in mitochondrial electron transport and how they're similar and have homology with some complexes, but not all in chloroplast electron transport. So I've got two pictures that we have here. The first one, again, since we've finished mitochondrial electron transport, should look pretty familiar. And then I've got a new one, and don't worry, I did this intentionally. It looks like this is backwards, upside down, all of that. But what I really have here is I really wanted to be able to compare these. So in order to do so, I want to have sort of the matrix be equivalent just in physical space to the stroma, which is why I have this guy flipped upside down so that we can sort of see that piece. So that's why that's flipped upside down, so we can sort of have this direct comparison as to where things are in relationship to matrix and stroma versus intermembrane space and intermembrane compartment. Okay. So let's start over here first with mitochondrial electron transport, since hopefully that's familiar, having been a learning unit that we've completed, but highlighting where we start with electrons is they come in either as NADH from entering in at complex 1, or FADH2 entering in kind of as part of complex 2 being succinate dehydrogenase. So let's kind of walk through the electron path, if you will. Okay, so we've got NADH. NADH is going to become oxidized, give up its electrons, becomes NAD+. And the key here is that we are going to return for more electrons from fuel oxidation. So return for more electrons from fuel oxidation. So let's walk through kind of what's happening here. Those electrons go from NADH, they get delivered to complex 1. Right? We can see that on the picture that we have up there. And we talked about the wiggling that happens with complex 1 to translocate protons to generate and contribute to our proton gradient. Electrons then are passed onto the mobile carrier Q. This is also the point where we're going to have electrons come in from complex 2, which again is FADH2. Once it becomes oxidized, it becomes FAD. It too will go and return to kind of get more electrons. And it's not necessarily that it really returns. It really just continues to do its succinate dehydrogenase chemistry and generate FADH2. But now from Q, we're going to deliver electrons to complex 3. So that interchange between electrons being given to Q and Q picking up protons from the matrix and contributing to the Q cycle, right? That's how Q and complex 3 kind of here contribute to the proton gradient. Then we have delivery of our electrons one at a time to cytochrome C. Okay, then cytochrome C is going to deliver them to complex 4. Final electron acceptor is molecular oxygen, generating water. Okay, so that one's pretty straightforward, stuff that we've learned about. The key here is I want us to start thinking about the energetics of this. So I'm going to draw an energy diagram here that basically is going to plot energy, okay, as a function of progress through the electron transport chain. And we're going to make sure we highlight that this is in mitochondria. So electrons come in high energy by virtue of their chemical composition being part of NADH. And when they ultimately get delivered to molecular oxygen and make water, those electrons are fairly low in energy. So what we really have is a downhill slide here to start from a fairly reduced molecule, NADH, very high energy in terms of its standard reduction potential. Okay, FADH2 is going to come in a little bit further down the path here. But the whole intent of looking at a diagram in this way is any time that we are going downhill in energy, that means we can use that energy to synthesize a proton gradient. And just reviewing, we've talked about several ways that happens. In complex 1, that's through the wiggling that happens, conformational changes in complex 1 that allow for pKa shifting to both pick up and then let go of protons, picking them up from the matrix and then delivering them to the inner membrane space. We talked about the chemistry that happens with the Q cycle, right? Q is going to pick up its protons from the matrix, right? So when Q gets its electrons and it becomes QH2, it's grabbing those 2 protons from the matrix, so it contributes by pulling those protons out of the matrix, and then when it lets go of those protons, when it lets go of its electrons, it releases them to the inner membrane compartment. So even though they're showing it through complex 3 here, it's really Q that's doing a lot of that proton translocation, but it's through its Q cycle capabilities in connection with complex 3 that we're able to do that. And then the last way that we're able to contribute to the proton gradient is with complex 4. We've got 2 kinds of protons there, right? We've got the scalar protons. So when we take and deliver those electrons to molecular oxygen, it picks up protons from the matrix to generate water. So just like Q is picking up protons from the matrix when it picks up electrons, so too is oxygen. When it picks up its electrons, it's going to grab protons from the matrix. That contributes to the proton gradient by getting rid of protons in the matrix. Now, unlike the Q piece where we ultimately do deliver them over to the inner membrane space, that's not the case here with these scalar protons that we have with molecular oxygen picking up protons. But complex 4 also has what are called those vectorial protons. Not well understood, but we have some protons that are moved physically from the matrix to the inner membrane space. So that's one of those important things, is to recognize how these different complexes are able to contribute to our proton gradient, which is then used in complex 5, our ATP synthesis machine, to synthesize ATP as those protons are delivered back to the matrix space. Okay, so that was a lot, but hopefully a review because we've already learned about mitochondrial electron transport. So let's see now what happens and compare it to what we see in chloroplast electron transport. And it's going to be a little bit hard to sort of see the picture. Don't worry, there's a separate video here that goes through the walkthrough of the electron transport in chloroplast, where this is right side up so we can sort of see it. But I just kind of want to draw sort of an electron flow, if you will, here and sort of talk about what happens. Now, I'm going to start here with something called P680. So P680 is a chlorophyll that's found in chloroplasts. Okay, the 2 main chlorophylls that we're going to talk about are P680 and P700. Those numbers have to do with the wavelengths of light that they match or correlate with to absorb energy. So 680 nanometers and 700 nanometers. Okay, so this process sort of starts with this photosynthetic pigment, this chlorophyll, being oxidized or losing its electron. So it becomes P680 star. Okay? And so that electron is going to go and be part of photosystem 2. So that's sort of this first photosystem that we have here is photosystem 2. Okay, so what needs to sort of happen here is we need to replace that electron, and this is where the electrons really come from water. So we're going to be talking about here, this is water. Okay, so water is going to be oxidized. So we're going to oxidize water. We're going to generate molecular oxygen and then an electron. And that electron is ultimately what gets delivered to P680 plus, that cation, to allow it to go back and become its neutral state. So it's a little bit weird because we first eject an electron from the chlorophyll and then it's replaced from the electrons that we get when we split water and generate molecular oxygen. So it's a little bit like not a straight flow, if you will. So the electrons originate from water, but they initially are ejected into the electron transport chain by being ejected from that chlorophyll. So one other piece that's important to sort of highlight here is it's important that these electrons get excited. So I'm going to draw a little sun here because these electrons in the chlorophyll are not necessarily high energy. We're going to have another little video that'll sort of show how this process happens, okay? Because not every chlorophyll is giving up its electrons. There's many that are just harvesting light energy. But ultimately, we're going to have these electrons that are excited and high energy because of their interaction with photons from the sun. Okay, so that's important because this is going to be what increases their energy. Okay. Ultimately, after these electrons leave photosystem 2, they're going to go to Q. Okay, this is homologous to what we see when complex 1 gives its electrons to Q. And then Q is going to go give its electrons to something called cytochrome B6F. Okay, and when we do our chloroplast electron walkthrough, we'll do this again, but just so that we have it down here, I'm going to draw places where there's homologs. Okay, we also have a Q in mitochondria, so this blue reminds us that it's a homolog for what we see in mitochondria. We've got complex 3, which is homologous to cytochrome B6F in chloroplasts. Okay, after we sort of leave cytochrome B6F, we're going to go to something called plastocyanin. So plastocyanin is homologous to cytochrome C that we have in mitochondrial electron transport. Okay, and then those electrons are going to be delivered to photosystem 1. Now, we're going to need to see again this process where we have excitation. Probably could have just redrawn this. We're going to use the sun and we're going to increase the energy of those electrons one more time. And at the end of the day here, what's going to happen is we're going to deliver these electrons to something called ferredoxin. Okay, and then ferredoxin is going to deliver those electrons to a complex called FNR. And here is where those electrons are finally going to be delivered to NADP plus to generate NADPH. Okay, and don't worry if that's kind of complicated right now, and that's a lot because we really haven't done a walkthrough with electron transport yet. So maybe watch that video first rather than looking at this comparison. But what I want to make sure to do here is draw a comparison for what we're going to see from the energetics that we have here. So I'm going to slide this guy over just a little bit because I want to make sure to draw sort of a parallel here. Going to use green here to make sure to highlight that this is the energetic diagram that we have when we think about chloroplast electron transport. So this is going to be electron transport in our chloroplasts. Okay, so here's where things are. Remember, water is fairly low energy. So we're going to have water being a fairly low energy sort of source of our electrons. So we're going to have electrons. The net is there from water. And what ultimately happens is that we're going to be elevating them by exposure to photons from sunlight. Okay? And so they're going to become high energy in photosystem 2. So as they sort of get translocated, wants to make that a straight line there, okay? We are going to be able to generate a proton gradient as they go downhill in energy. So again, proton gradient. Let me move this down just a hair here. Okay, now what's going to happen is we are going to get to a point where we are in photosystem 2 and these electrons are going to get excited again. So photosystem 1. The reason that those numbers are backwards is they actually discovered photosystem 2 first. Or I'm sorry, photosystem 1 first, but it actually comes second in the sequence of events here. Okay, but ultimately we're going to kind of come down in energy again, and we're going to generate our NADPH, which is a high energy molecule, just like NADH is high energy. NADPH is high energy. And as we have these electrons kind of going through electron transport, we're able to synthesize a proton gradient again. So, and let's put our sunshine in here. We have to make sure that the reason these get to go high energy again is because of photons from the sun. So, this is what we call a Z scheme, okay? So the fact that we go up and then down, and then back up again. This is what we call a Z scheme. Okay, it zigzags back and forth. So chloroplast electron transport adopts what we call a Z scheme, going back and forth between low energy and then using light energy to excite it to a higher energy state, allowing those electrons then to transit through the electron transport chain complexes, generating a proton gradient, and then a second time in a second photosystem, we use, again, a different wavelength of light to then excite those electrons. So highlighting here, this first one, let's put in, this first one is going to be 680 nanometers. The second one is 700 nanometers. Different wavelengths of light operate those different photosystems. Okay? So if I just flip to another slide here, which has our same basic sort of information that we just worked through down on the bottom, this is just highlighting for us the same sort of thing here. Now imagine, here's our different complexes. Imagine kind of taking those and spreading them out, like moving complex 4 all the way to the end, putting them sort of in order. We can sort of see that, yes, we go downhill from complex 1 and starting with NADH all the way down to water here. And looking over on the right, we can see more of what we actually have represented with our picture here, that we can see that we start out with electrons that are fairly low energy because they're from water. And then we're going to need to use 680 nanometer wavelength light in order to excite those electrons. And then as they go through Q cycle and so forth, it's going to allow us to pump a proton gradient. And then we're going to have an excitation again in photosystem 1 here using 700 nanometer wavelength light, and then ultimately delivering those electrons to NADP plus to make NADPH in that FNR complex.